So for meiosis, meiosis in male and female, they're different. For female, meiosis begins when female was still a fetus. Female only make eggs when they were fetus. When they are born, the number of eggs that they have become fixed, become a constant, and they no longer make new eggs. And each month, so we call it gametocyte. Those are the diploid precursor cell when they were in fetus. But then the process of meiosis somehow stopped in the middle after they were born. And then, in other words, the number of eggs a female have, they are fixed after they're born. And what happens is that when the eggs is being fertilized by the sperm, then the meiosis will complete. So this is something you have to keep in mind, and you will see it again and again, is that the meiosis for female eggs, they started when they were fetus, and then when they are born, then the meiosis process stopped. And then when the egg is being fertilized, then the egg will complete the meiosis. So one gamete resume meiosis each month, and that is the period. And then, although we will resume the meiosis each month, but it is not complete until fertilization occurs. And then in male, it is the testosterone to induce the meiosis in the testes of the male during puberty. So men start to make sperm during puberty, and men continue to make sperm when they were very old. But for female, the gamete, the ability to ovulate generally disappear by the age of 50. We call that menopause. And then cell cycle. We need to know the cell cycle so that we know when the cell division occurs. Cell cycle can be as short as 90 minutes, but it can be 24 hours in most of the human cell. So cell cycle include karyokinesis, I already talked about this, and cytokinesis, I also talked about this. So the splitting of the chromosome is the mitosis, and the splitting of the cell creates two daughter cells. We can briefly take a look here. So there is something called interphase and mitosis. Most of the time, the cell there in the interphase. In the interphase, you have G1S, G2 phase, and also you have a G0 phase. So what are those? And then we will talk about each one, I believe. I think we'll talk about each one briefly. Yes, we'll talk about each one briefly. So in G1 phase, the cells are growing and they are ready to replicate DNA. In S phase, DNA replication occurs. S stands for synthesis. You may call it synthesis. So it is like during this phase, the DNA make itself, synthesize itself, so that it's like replication. G1 phase, G you may stand for grow, the first growth phase. And then in the S phase, DNA replicate. And then in G2 phase, it's like the second growth phase. And then in here, you have a checkpoint. You have a checkpoint here to make sure that before the cell goes into mitosis, everything is correct. And then if everything is correct, then the cell goes into mitosis. And then after mitosis, at the end, at the end of mitosis, it goes back to the G1 phase. During the interphase, which is the majority of the time the cell span, the cell can take in nutrient and make other substances. The cell can grow. The cell can perform its specialized function. And then also the cell will prepare to divide. And, G0 phase. What is G0 phase? So after mitosis, the cell goes back to G1 phase, and you have two identical daughter cells. And then sometimes the cell will go from G1 phase into G0 phase. What is that? G0 phase is a phase that is resting period, meaning that the cell goes to G0 phase and then they don't do anything. They don't grow, they don't replicate DNA, they don't do mitosis. They just stay there, perform their normal function. An example of the cell that spend most of the time in the G0 phase, they are the neurons, the nerve cells. They spend most of the time in G0 phase. So in G1 and G2 phase, the growth phase 1, growth phase 2, the size of the cell and the protein increases inside the cell. And also they have checkpoints there. They also have checkpoint in G1 phase. They also have checkpoint in G2 phase. Why? They have checkpoint in G1 phase so that make sure that the cell are growing normally before they replicate the DNA. And they have checkpoint in G2 phase because they have to make sure that the cell looks normal. The cell is normal before they go to mitosis to split into two different cells. So as I said, S stands for synthesis of DNA. Now this is important. This is the question I ask my physiology students, but many of them don't know the answer, is that how many chromatids after replication in a chromosome? So it brings up to another key question. The 3 chromatin, chromosome, okay? Chromatids. So in the cell nucleus, normally, most of the DNA, they wound up to be a chromatin. So I remember I gave you this. I wrote this on the board. So DNA, and then DNA wind surround some histone protein, and then they become chromatin. The chromatin continue to wind themselves in the secondary or tertiary level, and then they become chromosome. So that is the relationship between chromosome and chromatin and DNA. But what about chromatid? Chromatid is actually the arms. Oh, okay, it already tells you sister chromatids, so this is chromatids. So in other words, before replication, you only have 1 chromatids. After replication, you have 2 chromatids. So that's the difference. I asked this question to my physiology student, yeah. So make sure that you know this right now. Now G0 phase. I briefly mentioned G0 phase because it is a safe haven. It is different from, in G0 phase, no cell division, and then G0 phase lasts from a few days to the lifetime. That's why a neuron can stay in G0 phase for so long. So it gives you an example, liver. Liver cells stay in G0 phase, but it divides once a year on average. But neuron, that is a nerve cell, neuron, they stay in G0 phase for their entire lifespan. So that's why I keep saying that nerve cell or neuron, they don't divide. So the gene for DNA replication and cell division, they are not activated during the G0 phase. If these genes, cell division, cell replication gene, they are activated, then they will prevent the cell to going into the cell cycle. I told you there are different checkpoints at different stages. So this is the checkpoint. This one checkpoint before the exit of G1. The reason is that they have to make sure that the DNA genetic material in G1 phase, they are correct. And there's another checkpoint at the end of G2 phase because they want to check the DNA information, whether or not they are correct before they go into mitosis to replicate itself. And those checkpoints we call it cycle regulatory protein, cell cycle regulatory protein.